Hello, everyone, and welcome to today's webinar, Oncomine Reporter, Integrated Reporting for Solid Tumor and Myeloid Somatic Variants. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. David Seidman, Scientific Director, Molecular Diagnostics and Serology Laboratories with Sentara Healthcare Laboratory Services. Dr. Seidman, you may now begin your presentation.
Thank you for the introduction, and thank you for the opportunity to present to you today some of the work that we conduct in our laboratory around somatic variant reporting and analysis. And specifically today, I'm going to be talking about our usage of Oncomine Reporter software and how we've utilized that in our laboratory for a standalone integrated reporting mechanism for some NGS profiles that we perform. Before we begin the presentation, I do have some disclosures here that are presented on this slide, just some standard ones. And with those, we'll move on. So first, I want to give a little bit of background about who we are at Sentara. Sentara Healthcare, we're a network of hospitals that are spread throughout the states of North Carolina and Virginia, predominantly located in the southeastern region in the Hampton Roads area, but spread throughout the entire state up to Washington, D.C., Western Virginia, and the northeastern corner of North Carolina. We have over 200 physician offices that are associated with us as a Sentara Medical Group. We're in a private payer insurance company as well. And our reference laboratory is located in the flagship hospital pictured here in Norfolk, Virginia. And this is where our hub is for testing and where our molecular laboratory resides. Within our laboratory, our annual test volume is over 9 million annual tests that are conducted in our integrated laboratory here in Norfolk. Of those, approximately 420,000 are molecular-based tests, and of those 420,000, approximately 1,500 are molecular oncology-based. For our molecular laboratory, we're spread across three independent principles for testing that we perform in the laboratory: molecular oncology, genetics, and infectious disease. Infectious disease, we perform a variety of testing, whether it's viral load genotyping analysis, infectious disease diagnostics, say for herpes or respiratory pathogen panels. We perform a significant amount of COVID testing and are currently looking into other viral testing for emerging infections that are currently going around. We also perform molecular genetics panels, carrier screening panels for cystic fibrosis, Fragile X, spinal muscular atrophy, perform factor V Leiden and prothrombin gene mutation analysis. But for today, we're going to be focusing on our molecular oncology work, particularly with our next generation sequencing profiles for solid tumor and myeloid analysis. But we do offer some single gene testing for solid tumor, EGFR, KRAS, BRAF, NRAS, and some myeloid heme testing as well for JAK2 and BCR-ABL. So in our laboratory, our molecular NGS workflow, and particularly I'm going to focus on our solid tumor workflow here. We perform about 700 solid tumor next generation sequencing profiles annually. So approximately half of all of the oncology work that we're doing in our laboratory is focused on this solid tumor NGS profile. Orders are submitted to our laboratory by an oncologist or through an in-house pathologist-driven reflex that we have within our healthcare system. Of this, approximately 70% of the cases that we receive are part of this non-small cell lung cancer reflex that is, again, pathologist-driven. And the goal of that reflex is to be able to provide an NGS report within 1 week of receipt of that reflex case. And so in our laboratory, at least historically, these cases have been analyzed in a week-long workflow where we're able to begin that process at the beginning of the week and return the report by the end of the week. Historically, in our laboratory with workflows that we've identified some bottlenecks within testing. So there's a lot of touch points or points within the process of performing next generation sequencing that can impede that time from sample to report. So with our prior workflow, or what I'm going to discuss as our current workflow on this slide, we had a semi-automated sample preparation system. We were preparing all of our NGS solid tumor libraries for sequencing manually by hand. And then our sequencing setup was a semi-automated sequencing that required a separate instrumentation for templating sequencing chip, and then that chip needed to be moved over to the sequencer. Post-sequencing, data analysis was a fairly manual process that we had employed. What we would envision as an ideal workflow would be one where we can automate that sample preparation and automate the library and sequencing as well as the data analysis with a manual review on the back end for the accuracy of those reports. So smoothing out that process or removing those impediments of sample to report analysis and allowing more time for our technologists to perform any extra data review or task that they may need to perform on a daily basis. So for NGS data analysis workflows, in our laboratory up until recently, data generated was analyzed across multiple different platforms and softwares. Each one of these required the transfer of data from one platform to another, and these can be significantly large files when you're talking about the sequencing output files. So just the single transfer of, say, a BAM sequencing file from one platform to another can take a significant amount of time. And if you're talking about caseload or for the week, that transfer could be a significant and slow the time to generating that final report. Additionally, when you're working across multiple platforms, each of those must be vetted and verified for their accuracy. So going between each one, verifying that the data transfer was appropriate, that the variants that were identified in one are carrying over to another, and that the content is correct. And so as mentioned, from the beginning of data analysis, in our laboratory, where that would begin with base calling, read alignment, all the way up to report generation, this data could have been transferred up to 3 or more times just to get to that final report. And so this has been a bottleneck for us historically in our laboratory is once we generate that data, how do we get that to the final report? And for a visual here, in our laboratory, previously we were using Ion Torrent Suite, where we could perform primary data analysis, overall run quality metric review, sequencing data analysis, and then transferring that large BAM file over to a secondary platform or software, Ion Reporter, where we would perform more in-depth analysis of the variants that were identified or mutations that we identified by NGS. And once we were able to perform that secondary analysis in Ion Reporter, we were transferring a VCF file over to a tertiary reporting software where we were generating final reports for those variants. And then finally, we were downloading a PDF file that is printed and reviewed by a medical pathologist or a director for accuracy, and then that was transmitted over to our LIMS system by physically scanning that PDF report back into the system. So a lot of processes that required either manual intervention by a technologist to move a data file or to get that report back out to the clinician. So now that we've talked a little bit about the background of our laboratory and testing, I'm going to talk to you today about our updated methodology or updated workflow for data analysis, and that's using the Genexus system and the automated next generation sequencing workflow to perform that analysis. So in the Genexus system, there's the opportunity to utilize an automated extraction platform pictured here on the left side of the image, the GPI, Genexus Purification Instrument. This allows for automated sample preparation, sample input, and then nucleic acid extraction output on the back end that's quantitated and qualified. It can produce data files that can be transmitted over to the Genexus sequencer pictured in the middle, containing all of the sample information that can then be programmed in for runs. On the Genexus sequencer, this provides us with the opportunity to automate our library preparation, our templating, and sequencing on board of a single platform. It requires very little hands-on time. The loading of nucleic acid on board the platform along with prepackaged reagents or primers. And then it allows for just walk-away time where the automation takes place for all of the preparation. And depending on the number of samples that are being run that day, the whole process takes anywhere from 18 to 26 hours. So if you factor in sample preparation and then sequencing, in as little as a day, we can get results back for sequencing results that we can then go over to reporting. And so on the final column, and where we'll focus today, is our reporting mechanism or data analysis mechanism that we're conducting within the Genexus software or Oncomine reporter. So in the Genexus software for data analysis, when we are logging in or to our account or to our sequencer, we're able to review overall quality metrics of the run. Here in particular, we're looking at a full chip run. Each chip has 4 lanes that are pictured over here on the right in red. For those, we can examine, how well our sequencing chip was loaded. In this case, about 86.39%. We can look at our total reads, sequencing reads, 47 million, our accuracy, our final reads of the library to get an overall picture of how well this sequencing run progressed and utilize these quality metrics to determine the likelihood of success of this run. Additionally, within the Genexus software, for the assays that we have developed here for our laboratory, we're able to check the QC status of each one of these samples prior to technologists logging in to actually review those. So within each pipeline, we programmed in a particular set of QC statistics that allow our technologists to just simply log in and see red light, green light almost for quality control for that sample. So green check mark indicating that all of our quality metrics have been met, an X indicating that there might be a particular quality metric within there that needs additional review or requires repeat analysis. When we click on that sample for review, we can see in this particular case that our sample QC for RNA has failed, particularly in this case, read length for that RNA wasn't sufficient. It would indicate to our technologists that this either requires additional review from the data quality variant review perspective, or that we need to go back and resequence this sample. But it's a real quick mechanism for our team to be able to understand which samples passed all of our overall sequencing quality control metrics and which ones may need that additional review or repeat analysis without having to dig deep to find that data. Additionally, within the Genexus software, when we are reviewing those, each one of those samples, under the Summary tab, we are presented with, say, variants that could be applicable for the type of sample that we're performing. Here on the Overall Summary tab, we're presented with variants that were identified in this particular case, which is a combination of samples. Here we're looking at a case that has EGFR T790M L858R variant, EGFR amplification, and then a MET fusion. If we wanted to see more details about these variants, we can click on the Variants tab located at the top and review our SNPs, our indels, and our fusions, our copy number variants by clicking on each one of the individual tabs at the top of that variants section. So in this, we can see, again, our T790M L858R variant, but we can also see more details regarding this. We can see the actual nucleotide change. We can see the chromosomal coordinates, as well as the effective read depths, or how deeply did we sequence each one of these variants. And we can use this to provide more confidence in the calls that we are generating. Similarly, we can click through the additional tabs. Here we're looking at the fusion in this case. And in this case, we can see that the MET fusion that was identified is an exon 14 skipping variant. And we can see that we have sufficient read counts for our determination within the laboratory to proceed and review additional items here, such as if there's any additional breakpoints or isoforms that may have been identified. But once we've gone through our base data analysis, our overall QC for the sequencing chip, our overall QC for each sample, and performed variant review, now we're ready to move into reporting, or how we would report these variants within our laboratory. So with the workflow that we have developed at Sentara in our laboratory, we've employed both the Oncomine reporter as a standalone version and the Oncomine reporter software as an extension. Neither of these softwares come preloaded on the Genexus platform. It's the end user's choice to download and utilize these softwares. The standalone does go on board the Genexus platform and can be logged into by a separate IP address that's kind of partitioned from the main Genexus platform. And then the extension can be downloaded directly into the Genexus software. And again, these are, I guess, platforms that we have elected to utilize. But first, I'm going to discuss our usage of the Oncomine reporter as a standalone platform or software. So when we log into Oncomine Reporter, we're presented with the landing screen where we can sign in. And once we have signed into Oncomine Reporter, we land on the splash page where we're presented with a couple of options here. At the top, we can see that there is a couple of icons that we can click and drop down on, whether it's analysis, settings, account. Under the settings heading, there's a couple of things that we can modulate or modify that'll customize for our laboratory. And so at Sentara, we have taken, say, and made a custom report template, which I will display later. And that takes a little bit of time to create within the software, but once you get comfortable with that generation, you can generate a report template that's customizable for your lab and your assay within about 5 to 10 minutes. You can also modulate filter presets for how you'd like your laboratory to filter for clinical trials or therapies. You can program in your specific assay that you're running in your laboratory, or what genes you're evaluating. So if you're running a DNA panel, you can program in what your DNA panel is, what specific genes you're targeting. Similarly, if you're covering fusions, RNA panel, or copy number variants, that can be added there and customized to your liking as well. Additionally, there are other options there for cancer type findings, biomarker descriptions, tier classifications, and signatory roles that can all be modified by the end user. And we'll talk a little bit about the signatory roles later. But once you've configured Oncomine Reporter to the likings of your laboratory, once you log in and you've got it configured, it's as simple as clicking on Create Analysis to begin generating reports. And once you click this icon here, you're brought to a page that starts that analysis portion. You can select your VCF file. In this case, we would still need to download a VCF file and upload it into the software. But once we've uploaded that VCF file, we give our analysis name, we select our filter preset that we've set for our laboratory, and then we select our assay. And you can see, again, it indicates the genes that were assayed within the particular VCF file that we're uploading. So these are all things that we can modulate ourselves. Once we've done that, we're able to upload and review content. So the VCF will be analyzed. And as long as it took for me to transition that slide is how quickly we're able to begin reviewing the context of the variants that were identified in that VCF file. So here in this particular case, we've identified a KRAS G12C variant. We can see on the overall summary tab that there are some biomarker-based relevant therapies that have been assigned, a number of clinical trials, allelic frequency, prognostic significance, etc. There's relevant wild type findings that are currently grayed out, but if we wanted to, we could elect to check those boxes to include them on the final report to place them in context. And then at the very bottom, we can see there's some additional information, whether that's regarding, say, a therapy or trial that might be contraindicated, a breakthrough, or fast track file. Overall, on the summary page, if there was something that we wanted to, say, include or exclude, we have that option as a laboratory. So say if we did not want KRAS G12C to show up on this report, we could simply uncheck that box next to the variant. Additionally, we can dig in a little deeper on each variant if we elect to, and select the Details tab here. On this Details tab, it will pull up a more in-depth review of the particular variants that we've identified. So again, looking at KRAS G12C, we can see the tiering classification again at the top as 1A. If we decided that, in this particular case that the G12C was not a 1A variant, we can modify that. We can also exclude it from key variants on our report. If we still wanted to include it, we could demote that variant by clicking on the Exclude from Key Variants icon and make it a non-key driver variant that would still show up on the report, but not in the context of being a key driver variant. Additionally, we are able to review in more detail each one of these calls, so the tiering criteria that met. Why was this classified as a 1A variant? We can review the FDA-approved therapies that are being presented to us as a laboratory. So here we can, again, see two specific types of therapies that have been presented to us. We have variant-specific therapies that are KRAS G12C specific or KRAS G codon 12 mutation specific, and then also non-specific therapies that are more at the gene level, so just KRAS mutation. And for each one of these, we can elect or select to include them on our report or exclude them. So say in the context of a variant-specific therapy, if we determine that the contraindicated therapies listed for KRAS G12 and colorectal cancer may not be applicable for a non-small cell case, we as a laboratory can make the election to exclude those from our report. Similarly, if we don't want to report gene-level therapies, we can deselect those as well, or therapies that we think may not be relevant. So we can see those are for endometrial or colorectal. We can expand each one of these icons or subsections below NCCN guideline, ESMO, clinical trials, and review the context and criteria there for each one. And similarly, as we have described for the FDA-approved therapies, we can go through as a laboratory and modulate those ourselves and make the final selection that we feel comfortable with as a laboratory for each one of these cases. Once we've gone through our summary or in-depth detail on a particular variant, the next step is to generate a report. And we click on the icon at the top. And once we get to the Generate Report tab, we will select our custom report. So we have a couple of Sentara-based report templates that we utilize. And on that Sentara report, some of the things that we've made that we can do within the software is we can add free text to those reports. So we can add our internal case information, a little bit more information about the tumor, such as the pathological review of the tumor content of the slide in a percentage format, who our clients are, physician, ordering pathologist, and what our internal container ID information is. And so we can customize this report and custom tailor it for each case that we have. So not only using the custom overall report, but when it comes down to each individual sample, we can tailor that report at the individual sample level as well. So once we've completed this information, our report is ready to go, and we just click Download Report. And this is how it would come out on the back end. So this is an example of a customized report that we have for the non-small cell case we've been going through. Our relevant findings for non-small cell are presented here at the top for genes that were assayed. We see the KRAS G12C being called out overall, and then not detected in the remaining genes in that panel. We see our relevant biomarkers table, which gives a little bit more information about that KRAS G12C, the allelic frequency, the variants that were identified, any breakthrough or fast track alerts that are below. Those are signaled to the end user of the report. And then on subsequent pages, we do provide more information about the variant, whether it's the specific read depths or quality calls, information about trials or therapies as well. On the final page or near final page of the report, we have our customized assay that we're presenting, the genes that we tested for, any disclosure statements that we've elected to include on the report, methods and limitations, a laboratory statement, and then finally, we have a location where a medical director or a pathologist can review and approve of this report. And so once this report has been generated and printed, it can be signed out and scanned back into our LIMS system for reporting. However, another feature of the standalone Oncomine reporter system is that once this report has been generated and reviewed, we have the opportunity to place an electronic signature on that report. So we can assign signatory roles within the Oncomine reporter software for different levels, whether it's a technologist reviewing the case or it's a, say, pathologist who is reviewing and signing that case out. And so once we have gotten through the process of reviewing that, we can create logins based on signatory roles. A sign report icon will populate, and once we click on that, it will require a password for the individual who's signing that report, what role they're signing under. Here we're displaying, say, a medical director for Sentara Molecular Diagnostics, any comments that they would like to add, and then they can just click Sign Report. And in the end, their electronic signature will display in the Oncomine reporter software. You can see all of the fields that we had previously been able to edit, such as our template or case information has now been grayed out and cannot be modified. So once this report has been signed out, it is finalized, and no additional edits can be made unless the signature is removed, and then we can go back and make modification. But once the electronic signature has been applied, we can download that report to review it again, and we can see our medical director's signature has been electronically applied to that report. And we can go ahead and either print this report out and scan it into our LIMS, or another option that we've been able to work through with the Oncomine reporter software is the ability to send this report. So this icon here, we've been able to configure with our LIMS system to transmit this report directly from Oncomine Reporter into our LIMS. So it allows the electronic transfer of that final report with the electronic signature over and doesn't require any download or printing or manual signatures of that report. It also helps ensure that the correct report is going into the correct location within the LIMS system. If you take into human factor and seeing those reports get scanned into the LIMS, there can always be a chance of error of that report getting scanned in under the incorrect specimen. This helps prevent that potential error step or pitfall by sending it directly back to the container ID that we have programmed within our system. So this has been an overview of the standalone Oncomine reporter. So again, this is something that we've downloaded and we utilize as a standalone configuration within our laboratory. There's also the option for working within the Genexus software with Oncomine Reporter extension. So here you can see under our setting configuration, we have an Oncomine Reporter selection. And again, this is a software that we have downloaded within the Genexus platform. It does not come preloaded with Oncomine Reporter. It would be something that would be the laboratory's choice to download and employ this within their lab for usage. But if we do click on this settings icon and we do want to go into Oncomine Reporter within the Genexus software, we would just click on that icon there. And similar to the standalone model, we're presented kind of with a splash page, except this one is a little bit reduced. And what you'll see is instead of seeing the overall screen where you're presented with an opportunity to, say, create analysis, we're more looking at the settings of the software here. So report templates, again, filter presets, biomarker descriptions. These are things that we can custom tailor to ourselves within the laboratory, and you can see that we've created a few separate report templates for our own usage here. Sentara report template overall, one where maybe we're only running a DNA-based panel report template, one where we might not be reporting copy number variants due to a quality metric that maybe wasn't met or a low tumor percentage. But we're able to manage those report templates and customize them for our needs, whether it's on an overall basis or a sample-based analysis. And again, once you've created your report templates, you're ready to go with analysis within the platform. So within, again, the software, if we're looking at our variants tab where we were performing review earlier, in this case, we're looking at myeloid malignancy where MIPL 515 variant was identified. What we can see once we have the Oncomine Reporter software installed is that there's an icon that we can click on that will launch Oncomine Reporter out of the Genexus software. Clicking on this icon, it'll pop up that review tab that we can see that we're looking at in the standalone version earlier. We can review criteria, again, by clicking through these different tabs, summary, details, or non-key variants that may have been identified in this case. And then we can, again, see the overall summary review for tiering, allelic frequency, or any prognostic or diagnostic significance. Similarly, within the extension version, we're able to go through and review each one of these calls at a more detailed level by selecting that Details tab. Again, we can expand to review the tiering criteria that was met. We can review clinical trials. In this case, there's no therapies that were identified or approved, but we can again go through and perform that review and determine if this meets our needs and are we in agreement with the call that is made here. And again, we have the option to modulate this as needed. One tab that I haven't gotten into just yet is the non-key variants tab. And on this particular case here, we're looking again at a case where a RUNX fusion was identified. Similar on the summary tab, we see that has been identified with the similar findings of other variants that we've discussed earlier with the biomarker-based therapies or prognostic clinical trials, etc. But in this case, we did identify a non-key driver variant. And if we click on this tab, we can see that listed here. This B-core cysteine 94 tyrosine variant at a lower allelic frequency, 6.1%. If we click on this tab and we see that there's a variant here, or we've identified it previously through our data analysis, we can perform a deeper review of this variant and determine, is it applicable to the case that we're profiling? Is there anything of significance that this might be worth reporting out as a relevant finding on the case? And if we determine internally that this is a worthwhile variant to include as a relevant variant on the case, we have the ability to promote that. So similar to how we can demote a variant to become a non-key driver variant, which will still show up on the report. Just to be clear, the non-key driver variants do show up on the report in a section that is separate. But if we decided we wanted to promote this, we would just click the Actions button here, and we can promote that to a key driver variant that will show up at the top of the report. So if we've decided that this B-core variant should be reported out, we have the ability to promote that and include it on the report. Similarly, if it's something where we feel like maybe this B-core variant isn't really significant, we don't want it on our report at all, we don't want it in the non-key driver variant section, we don't want it on the report at all, after we promote it, we can deselect that box and it will not show up on the report at all. So it really leaves it to the end user to be able to make those calls and modifications to their final report. But if we were okay with this being the report that was going to go out, the next thing we would do is we would save that, and then we would click over onto the Reports tab in the Genexus software. So we went through a few of these different ones earlier where we went through the QC, the overall summary, the variants, and now we're looking at our Reporting tab. In this Reporting tab, if we click Generate Report, it'll pop up a screen that allows us to select Oncomine Reporter Report with the software we've installed. It allows us to select our report template highlighted there as our Sentara report template. And when we do that, our customizable fields that we have elected to include on our report pop up. And we're able to complete these to our needs. Once we've done that, we click Generate, and we have our final reports that we're able to download or utilize for our needs. So we have our NPL 515 case on the left, our RUNX fusion on the right. Similar to our solid tumor profile that we displayed earlier, relevant findings shown at the top of the report, genes where we detected particular variants, genes that are relevant where we did not detect anything, the findings from the relevant biomarkers, whether there's any trials or therapies associated, prognostic significance, and even diagnostic significance if we're including that on our report. But this would be the final report that we generated, and this was all done within the Genexus software. So we didn't need to transfer a data file anywhere. Once we've downloaded the Oncomine Reporter software into Genexus, it becomes integrated into that workflow where there's no transfer of large data files like the BAM file. The VCF file is self-contained within the platform, and we're able to get a report. So it really reduces that need to transmit these files across multiple different platforms just to get to this point where we have a nice, what we feel is a nice clean report on the back end to provide out. So in conclusion, the Oncomine Reporter software can be provided as a standalone software that is partitioned from the overall Genexus platform or integrated as a kind of additional widget or software within the Genexus platform. It allows for lab-specific customizable reports that are tailored to the needs of the assays and panels that we're performing. The hands-on time, which has been pretty significant in the past, is significantly reduced with the Genexus workflow, particularly with the data analysis. I think as mentioned with the integrated workflow, all of that data analysis can be contained within one platform, from raw data analysis, overall quality metric, variant review, down to the report generation. In standalone mode, even with it being separate, it only requires the transfer of a VCF file, and at that point, it's a filtered VCF file, so it's very small. It's only the variants that we are looking to report. Again, as mentioned, the Oncomine extension can be installed by the end user, which requires no data transfer. The variant review and classification within the platform we feel has been significantly simplified. Whether you're working in the extension or the standalone, the report generation does not take a significant amount of time. It's only a few clicks. It doesn't require navigation to a few different pages or a ton of input from the technologist to get to that final report. And then a feature of the standalone version is that electronic signatures can be applied. If a pathologist or signatory role wanted to log in electronically to provide that signature, they're able to, and we're able to transfer that data directly electronically from our LIMS system instead of needing to, say, print these reports, sign them, and then have them scanned back in. So overall, we find that the workflow, whether standalone or integrated, has presented us with a much better, smoother workflow for our laboratory operation. And with that, I'd like to conclude my discussion of the Oncomine Reporter software and its usage within our laboratory at Sentara, and I'd be happy to address any questions that you have. And again, thank you for your time.
Thank you, Dr. Seidman, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. And our first question is, how many variants do you typically have to curate, and how much time per report is required when using Oncomine Reporter for variant review and clinical curation of solid tumor samples?
That's a great question, and I think my answer is it really depends, and a lot of that is on a variety of factors, depending on the number of variants that you identify within your panel. I would say for us, we're using smaller, more targeted, focused panels, typically identifying a handful of well-known variants. So the time to review and report doesn't typically take that long. If you expand the content of your panel to a larger, maybe more expanded panel, or if you're identifying variants that may not necessarily be as well as classically known or defined, the length of time to review those can be extended, but that's really at the discretion of the end user and how in-depth that they want to go with each variant that is reviewed or curated. So again, it just depends on the content of the panel that you are sequencing and reporting, and again, if you're identifying, variants of unknown significance, tier 3 variants, or any other variant that might require that additional review, it's really at the discretion of the end user. But for us, if we have a negative report, it's very quick to generate that, from the time we upload the VCF, about 30 seconds to the report. If we're identifying well-known variants, maybe a minute or two, 5 to 10 minutes at the longest.
All right, thank you. Next question. How long is a typical Oncomine Reporter report?
Again, yeah, so another great question. Yeah, and again, this also depends. The report is completely modifiable. So any section that we choose to include on the report or exclude can either reduce or add to the length of that report. For us, a negative report would be about a page or two. For one with a variant that, say, has signed clinical trials or therapies, those can extend a little longer, anywhere from 3 to 11 pages. And those are just for the reports that we're generating. If you add additional sections in there, I think one of the sections that we don't include on our report are gene descriptions on that report. There's things that'll add to the length. So if you're looking for a lengthier report, you're able to make that. If you're looking for a short, compact report that can go out, you're able to achieve that as well. But overall, the reports that we're generating out of our laboratory are relatively, I would say, relatively compact.
Thank you. Next question. How often does Oncomine Reporter update content?
So for us, at least what we're seeing currently is that we're getting updates on a monthly basis. We're seeing those come through for the standalone and the integrated versions of the software. If configured within the Genexus system, those updates can be as simple as an automatic, I don't want to say automatic, but clicking an icon within the software to update that content. When they do update the content, they provide all of the updates and changes that have been made there, whether it's anything with the variant classification, any additional guideline recommendations or therapies trials, update to genes available, gene descriptions, etc. So it's a pretty comprehensive update packet that is provided depending on the extent of the update, but all that information is available there, and it's typically on a monthly basis.
Right. Thank you. Next question is, can we use customized panels from third parties, or does it have to be through Thermo Fisher?
So from my understanding, for the usage of Oncomine Reporter, the VCF file that is generated needs to come through either the Ion Reporter platform or the Genexus platform. So whatever data that is being generated, in order to push that through Oncomine Reporter, would have to be analyzed in either of those platforms.
Thank you. Next question. Can someone with a basic knowledge of bioinformatics utilize this platform for data analysis with ease?
I would say so. I think, within our laboratory, we have trained bioinformatician, but for the rest of us, none of us have that classical bioinformatics background. And we found that as a team that we're readily able to navigate these platforms and softwares without needing to have that necessarily in-depth background education, but it's a fairly straightforward platform to utilize.
Next question. How does the Oncomine reporter get access to what is to be analyzed?
So for our needs within our laboratory, we're downloading, if we're talking about the standalone version, we're downloading a filtered VCF file from the Genexus software from our data analysis. So that VCF file that's generated within the platform is then uploaded into the software. If we're talking about the integrated version, it's utilizing the data that's currently within the platform. So when we want to move forward or advance with a report using that integrated mode, all the data that's needed is pulled directly from the Genexus platform. So standalone, for us, we're downloading a file and uploading. In the integrated mode, it's simply having that, making that choice or election to install the Oncomine Reporter software there, and then once that's enabled, navigating through the software to have that data report generated.
Thank you. And we will wrap up with that question. Do you have any final comments for our audience?
No, just thank you for attending today. Thank you for your time. I really appreciate it and hope I was able to provide you with some good information or details about Oncomine Reporter.
Well, thank you again, Dr. Seidman, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.